Hello to all of you. Let's discuss chapter number 16. Chapter number 16 is about carbohydrates. And let me go to the slideshow mode. So, this chapter we are going to talk about carbohydrates. So, basically, in this course, we are moving towards metabolism. So, we have discussed few macromolecules, we have discussed nucleic acids, we have discussed lipids, we have discussed proteins. So now we are actually moving towards metabolism, and whenever we discuss metabolism, we usually start discussing how the food, so basically metabolism involves the food we are eating, how it gets digested. So the metabolism basically involves both catabolism and anabolism. Catabolism involves the breaking of the bigger macromolecules into smaller subunits, and there is energy released, and that energy is used for doing various activities by the organism, as well as there are various activities going on at a cellular level for which energy is required. And anabolism is basically building up processes, building up mechanisms, like the smaller subunits, which are the result which are produced in the catabolic reactions, like proteins get digested and amino acids are formed. In the anabolic reactions, these amino acids are together again and they form proteins. So these kind of processes, they need energy, and these amino acids are converted into proteins, or simple sugars are converted into polysaccharides, or nucleotides. They make nucleic acids. So why anabolic reactions are going on? Because our body needs to make more cells, and these are the components of the cells. So whenever we talk about metabolism, we always so basically when we're talking about catabolic reactions, breaking up mechanisms or of the food and the production of energy, we always start talking in back to glucose. You will see in this lecture, in the next chapter, we will be talking about glycolysis. So in glycolysis, we talk about the journey of glucose molecule. So how does glucose molecule gets converted into pyruvate in glycolysis, and some energy is released, and then followed by what happens to pyruvate, what other reactions it goes through, then it goes through Krebs cycle, and then oxidative phosphorylation. So this is whole big process which is called cellular respiration or aerobic respiration, and it is the process how energy is released from the food. So whenever we start talking about metabolic reactions, especially the catabolic reactions or the cellular respiration, we talk with taking one molecule of glucose. So what is glucose? So it's a kind of a carbohydrate. So in this is the macromolecule we need to learn in detail. So carbohydrates, they are of different kinds. They are monosaccharides, disaccharides, trisaccharides, or we can call them oligosaccharides and polysaccharides. So in this chapter, we are going to focus on carbohydrates. So first of all, why they're called carbohydrates? Now, what is the basis of the name? Carbohydrates. You can break this word. Carbo means from carbon, and hydrates means water. If you look at the general formula, so carbohydrates, let me tell you, carbohydrates, they are of they can be of different kinds. They can be saccharides and polysaccharides, and monosaccharides are the most simple form of the carbohydrates. They are very simple sugars, and this is the formula for the this is the empirical formula for the monosaccharides. You can see there is a carbon and there is a water. Okay, so if there are 6 carbons, there will be double the amount of hydrogens and the same number of oxygens as the number of carbons. So this is the empirical formula. Like for glucose, glucose is C6H12O6. So it has 6 carbons, so it has 6 oxygens and 12 hydrogens. So basically carbohydrates have got the name because monosaccharides, they have this simple empirical formula. So you can see on the base of this formula there is a water molecule attached to the carbon, so that's why they're called carbohydrates. Now, we find carbohydrates at polyhydroxyaldehydes or polyhydroxy ketones. So I'm going to discuss what ketone group is, what aldehyde group is. We can also define carbohydrates like that. There are three different kinds: monosaccharides, oligosaccharides, and polysaccharides. The word saccharide comes from sugar. Mono means simple sugars. Oligo means they have few monosaccharides which are linked to each other, and polysaccharide means there are multiple monosaccharides linked to each other. So monosaccharides are simple sugars, and this is the general formula for them. And oligosaccharides, they have few monosaccharides linked to each other by bonds called glycosidic bonds. And polysaccharides, there are multiple monosaccharides linked to each other by glycosidic bonds. Now remember that as we have discussed in proteins, we have discussed in nucleic acids, that these are big molecules, these are macromolecules, they are biomolecules, they are molecules of life. So they are polymers, and they are made of monomers. And how these monomers are linked to each other? These monomers, they are linked to each other by condensation reactions or dehydration reactions, where these monomers, they form covalent bonds with each other, and for the formation of the covalent bonds between each other, there is a loss of a molecule of water, and formed between these different monomers. These bonds, their nature is covalent bonds, which are very strong bonds, but they have different names in different macromolecules. Like proteins, the name is peptide bonds. Then in the nucleic acids, they're called phosphodiester linkages. In carbohydrates, they are called glycosidic bonds. Okay, the main functions of carbohydrates, as you know, sugars and all that we eat because they give us a lot of energy. Now, oligosaccharides, they can help in cell-to-cell interaction and also immune recognition. So basically, oligosaccharides, they are short chains of sugars. They can be either attached to proteins, and they are called glycoproteins. Their function is basically in cell-to-cell recognition. So these glycoproteins, which have these oligosaccharides on them, they are kind of a markers on the cells, and on the basis of these markers or the glycoproteins which has these oligosaccharides, the cells are identified, as well as the cells, they interact with each other. And polysaccharides, they can be there can be structure form of polysaccharides, or they can be storage form of polysaccharides, like starch, like potatoes, rice. They are very rich in starch. Now, what is starch? Starch is a polysaccharide. It is made of multiple glucose molecules. Glucose is a monosaccharide. Okay, glucose is monosaccharide. Then fructose is monosaccharide. Then if glucose and fructose, they combine, they form a glycosidic bond with each other. They form a disaccharide called sucrose, and sucrose is a sugar which we eat every day. Then starch is a polysaccharide. It's made of multiple glucose molecules connected to each other by glycosidic bonds. So starch is a storage form of polysaccharide in plant cells. Okay, and similarly, glycogen is a storage form of polysaccharide in animal cells. And cellulose is also a polysaccharide, but it is a structure form of polysaccharide which is present in the cell walls of the plants. And that is why the plant cells, some of the plants, they're very hard, they have a hard outer covering. It's all because they have the cellulose in their cell walls, and cellulose is a polysaccharide. And we'll talk about all these things in more details. The monosaccharides, they're basically the building blocks of carbohydrates. Now, they can be of two kinds. They can be aldoses or they can be ketoses. If the functional group present in the monosaccharide is aldehyde group, then the sugar is called aldose sugar. And if the functional group which is present in the monosaccharide is ketone group, then it is called the ketose sugar. Okay, if there is an aldehyde group, it's an aldose sugar. If there is a ketone group, there is a the sugar is called ketose sugar. Now, monosaccharides can also be defined on the basis of how many carbons they have. If they have 3 carbons, they're called trioses. If they have 4 carbons, they're called tetroses. If they have 5 carbons in them, called pentoses. If they have 6 carbons in them, they're called hexoses. If they have 7 carbons in them, they're called haptoses. So we are going to talk, we're going to take the example of trioses, which have 3 carbons in them. The examples of trioses are glyceraldehyde and dihydroxyacetone. So glyceraldehyde, it has an aldehyde group, so it's a aldose sugar. Dihydroxyacetone, it has a ketone group in it, so it's called a ketose. So here is the structure of dihydroxyacetone. So dihydroxyacetone does not have a chiral carbon. So I hope you remember what chiral carbon is. It's that carbon, or chiral carbon is also called asymmetric carbon, to which all the 4 groups which are attached are different. That's a chiral carbon. So in dihydroxyacetone there is no chiral carbon, so it does not have, it cannot form the stereoisomers. Glyceraldehyde, it has a chiral carbon, so it can form the stereoisomers or the mirror images of each other. So the 2 stereoisomers of glyceraldehyde are D-glyceraldehyde and the L-glyceraldehyde. Now these sugars, they can be represented in different ways. That's what we're going to discuss. Now, what are Fischer projections? They are the 2-dimensional representations of the 3-dimensional molecules. So 2-dimensional representations are called the Fischer projections. And the bonds, so when the bonds, they are written horizontally, bonds written horizontally represent bonds directed in the front of the paper in 3 dimensions, and the bonds which are written vertically, they represent bonds directed behind the paper in 3 dimensions. So this is the Fischer projections for the glucose and the fructose. So glucose has a aldehyde group, so it's a aldose. Fructose has a ketone group, it is a ketose. And in these Fischer projections, most highly oxidized carbon is written at the top and is designated as carbon number 1. Okay, so you can see here the most oxidized carbon is written at the top and it is written as the carbon number 1. So you can see that how the carbons they are written. Okay, so this is the most oxidized carbon. So that is written as the carbon number 1. So this is the most oxidized carbon. Okay, so this is the example of glucose, which is the aldose, and the OH group, which is so now glucose is the aldose, and it can exist in the form of stereoisomers. So if the OH group is on the right of the highest number chiral carbon, okay, now this is the chiral carbon, okay, this is not the chiral carbon. In glucose there are 4 chiral carbons, there are 4 asymmetric carbons. Now this carbon is not a chiral carbon, because this carbon is attached to 1 hydrogen and 2 oxygens. This carbon is making 2 bonds with the same oxygen, okay, and then so this carbon is making 2 bonds with oxygen, and it is making 1 bond with hydrogen, and the 4th bond it is forming with this whole group. So this carbon is not chiral carbon. For a chiral carbon is that carbon which can form 4 bonds with 4 different atoms or 4 different groups. Now this carbon is a chiral carbon because this is bonded with hydrogen, it's bonded with OH group, it's bonded with this group, and this whole forms 1 group. So this is a chiral carbon, this is a chiral carbon, this is a chiral carbon, this is a chiral carbon. Okay, if the OH, so this is the first chiral carbon. If the OH group is on the right side of the highest number, so this is the highest number chiral carbon, if it is on the right side, then this glucose is D-glucose. If it is on the left side, then this is the L-glucose. Okay, now in these sugars, cyclization can also take place. So cyclization of sugars takes place because of interactions between functional groups on a distant carbon, such as carbon number 1 and carbon number 5, to form a cyclic hemiacetal in aldohexoses. So hemiacetal is formed by the reaction of aldehyde with an alcohol. So let me show you here. Now this is alcohol, this is aldehyde. Okay, so this oxygen can form a bond with this carbon, okay, as a result of which the compound which is formed is called hemiacetal. Now what is happening here is when this oxygen forms bond with this carbon, when this oxygen forms bond with this carbon. So this carbon can form 4 bonds. Carbon is forming 2 bonds with oxygen, third bond it is forming with hydrogen, and fourth bond it is forming with R group. Now when this oxygen forms bond with carbon, then this carbon has to break one of its bond. So when the hemiacetal is formed, carbon breaks its double bond with oxygen, okay, and form one bond with this oxygen. Okay, so right now it is forming 2 bonds with this oxygen, so it's going to break one of its bond with this oxygen and it is going to form one bond with this oxygen. Okay, so carbon can form 4 bonds, it's all good now. But what about this oxygen? Because it was forming 2 bonds with this carbon, so it's still forming one bond with carbon. How it's going to be satisfying its electronic configuration? Because this carbon needs to make 2 bonds. So to satisfy its electronic configuration, this oxygen is going to form one bond with this hydrogen. Okay, this oxygen breaks bond with this hydrogen and it can form bond with this carbon. This carbon has to break one of its bond with this oxygen, only then it can satisfy its electronic configuration. So this oxygen breaks its bond with this hydrogen, makes bond with this carbon. This carbon breaks one of its bonds with this oxygen and forms bond with this oxygen. What about this oxygen? This oxygen also needs to make one more bond with another atom. So it forms a bond with this hydrogen. So that is why there's a OH group here. So this is the way cyclization of these sugars can take place, and that results in the formation of hemiacetal. And what is hemiketal? It is due to the cyclization, due to the interaction of carbon number C2 with the carbon number 5, and that results in the formation of hemiketal. And hemiketal is a compound that is formed by the reaction of a ketone with an alcohol. So coming back to hemiacetal again, let me show you here how exactly it's formed in the sugars. Now this is glucose. You can see this is carbon number 1, 2, 3, 4, 5 and 6. Now how the cyclization of glucose takes place? The oxygen which is attached here to carbon number 5, so this is your carbon number 5, this is this carbon, okay, to this carbon is attached your OH group here. This is carbon number 6 which is attached to carbon number 5, okay, this is what it is, it's here, right? So this oxygen, which is I just showed you here, this oxygen is going to form its bond with carbon number 1, and this is what is carbon number 1. So this oxygen which is attached to carbon number 5 is going to form its bond with carbon number 1. So when it forms bond with carbon number 1, it has to break its bond with this hydrogen, only then it can form the bond with this carbon, and this carbon has to break one of its bond with this oxygen, and this oxygen is then going to form bond with this hydrogen, and the OH group comes here. Okay, this is the way this glucose form a cyclic form of a sugar, and this is called pyranose. So pyranose is a cyclic form of a sugar which has 6 carbons in them. So this is the cyclic form of the glucose. If the OH group is present at the bottom of the plane of this ring, then this form of glucose is called alpha glucopyranose. But if this OH group is above the plane of this ring, then it is called beta glucopyranose. And these ring structures which are formed, they are called Hayforth projection formulas. So like these are Fisher projections. They're 2-dimensional, okay, and these are cyclic forms of these sugars. They're called Hayforth projections. Okay. And anomeric carbon is basically the carbonyl carbon. And this is the Fisher projections. So in the Fisher projections, if the OH group is on the right side of the anomeric carbon, if the OH group is on the right side of the anomeric carbon, then this is a alpha form of glucose. If the OH group If the OH group is to the left of the anomeric carbon, anomeric carbon is a carbonyl carbon basically. If it is on the left side, then it is the beta form of glucose. So glucose has two forms. It exists as two stereoisomers, D-glucose and L-glucose. And then it has two other forms, alpha and the beta form also. And what is a pyranose? I just told you, cyclic form of sugar containing a 6-membered ring. And similarly, we have a furanose. It is a cyclic sugar with 5-membered ring. Okay, so this basically shows you the Hayforth projections for the sugars. These are all cyclic forms. This is the furanose, which has 5-membered ring, and this is the pyranose, which has the 6-membered rings. So this basically shows you the Fisher projections, complete Hayforth and abbreviated Hayforth projections. This is basically different ways of this. So usually the cyclic forms of the sugars, the sugars form the cyclic forms usually when they are added to water basically. Okay, now what is glycoside? So the compound formed, what is a glycoside? It's a compound formed when a hemiacetal carbon reacts with an alcohol to give a full acetal. Now this is your hemiacetal. So this is your alpha D glucopyranose. Okay, this is alpha D glucopyranose. So this is the D form of glucose and this is the alpha form. Okay, because the OH group, so why it's alpha form? Because the OH group is below the plane of this ring. Now when this glucose, and it's a hemiacetal, when it is going to react with some alcohol, okay, then it is going to be converted into full acetal and it is called glycoside, and it results in the formation, you can see here, this is methyl alcohol. So when this methyl alcohol is going to react with the OH group of the sugar, they are going to form a bond with each other, okay, and this bond is called glycosidic bond. And how this bond is formed? So the OH group, which is here, and this alcoholic group, they are together going to lose a molecule of water, okay, they're together going to lose a molecule of water, H from here, OH from here, and that is going to result in the formation of a bond between this CH3 and oxygen. And that bond which is formed between this alcohol and this sugar, that bond is called glycosidic bond, and glycosidic bond is formed by the removal of a molecule of water. So furanosides, they are the glycosides involving furanose, which means they have the ring with 5 members. Pyranosides, they are the glycosides involving a pyranose, which means they have sugars with 6 members. Now glycosidic bonds, they can be, so these glycosidic bonds, so as I said, the carbohydrates, they can be monosaccharides, disaccharides, polysaccharides. So when two monosaccharides, they link with each other, they form a disaccharide. Like glucose and fructose, they link with each other and they form a disaccharide called sucrose. Two glucose molecules, they can also link with each other. And how they link with each other? By glycosidic bonds. And how glycosidic bonds are formed? By the removal of one molecule of water. That results in the formation of, when two glucose molecules, they link with each other, they form a maltose. When one glucose and another monosaccharide called galactose, they link with each other, they form a disaccharide called lactose. So these glycosidic bonds, they can be present in disaccharides, they can be present in trisaccharides, they can be present in polysaccharides, because they are the bonds which are connecting them. And they can also be present, they are also present in polysaccharides. And the glycosidic linkages or glycosidic bonds, they can be alpha or beta. They can be alpha or beta glycosidic linkages. If the glycosidic linkage is formed involving the glucose, which is alpha glucose, then the glycosidic linkage is called alpha glycosidic linkage. If the glycosidic linkage is beta glycosidic linkage, it is because the glucose which is involved in the is the beta form of the glucose. Now you can see that these are glucose molecules, and they are these are two glucose molecules, and they are alpha form of glucose molecules. You can see they are connected to each other by alpha glycosidic bond. So 1 to 4 means this is the carbon number 1, okay, and this is the carbon number 1 of first carbon. One second. Yes, this is the carbon number 1 of this glucose, and this is the carbon number 1 of this glucose. So this is 1, 2, 3, and here is the fourth carbon. Okay, so these two glucose molecules, they are linked by alpha 1 to 4 glycosidic bond, which means this glycosidic bond is linking carbon number 1 of this glucose and carbon number 4 of this glucose. And this is alpha 1 to 6 glycosidic bond. So here the linkage is between carbon number 1 of this glucose and carbon number 6 of this glucose. And similarly, they can be beta glycosidic bonds also. Beta glycosidic bonds will be formed when the glucose molecules, they are beta forms. When the glucose molecules involved are beta form of glucose. The glycosidic linkages, they can be linear, they can be branch. You can see here like these are all linear glycosidic linkages, and there can be branching also, which means this is a chain of glucose molecules connected to each other by glycosidic linkages. They're all linear bonds. Okay, there can also be a linkage like this, and these are called branches. Okay, so this glucose getting connected to this glucose connected to this glucose. So these are branch points. Now, a very typical example of polysaccharide is starch. Now, starch can exist in two forms. It can be a amylose and a amylopectin. Amylose is actually doesn't have much branching in between, but amylopectin is a form of starch which has some branching in between. Okay, so amylase is amylase is an enzyme which can actually break the glycosidic bonds which are present in the starch. So amylase, they can be alpha and beta amylase, and they can break the alpha 1 to 4 linkages. So beta amylase is a exoglycosidase, which means it can cleave the glucose molecule. It can break the glycosidic bonds between the glucose molecules from the end of the polymer. And alpha amylase is an endoglycosidase, which means it can hydrolyze the glycosidic linkages anywhere along the chain. And there are some other enzymes also, they're called debranching enzymes, and they can break the alpha 1 to 6 linkages. Now, as I said, starch can be of two kinds. It can be amylose and amylopectin. Amylose is not much branched. Amylopectin is quite branched. Okay, and here we just talked about the enzymes which can break these kind of glycosidic linkages. Now, glycogen is storage form of polysaccharide. So just like starch is a polysaccharide which is made of multiple glucose molecules, similarly glycogen is also a polysaccharide which is made of glucose. And in starch and glycogen, the form of glucose is form. And glycogen, just like amylopectin, is highly branched. And glycogen is a storage form of polysaccharide in animals. Like in us, like if you eat a lot of glucose, okay, your body doesn't need that much glucose. So what is your body to do? Your body is going to convert glucose into glycogen, and it is going to store it in the liver. And when you don't, if you have not eaten like food for some time, you did not have the intake of sugar, and your body is going to convert this glycogen back into the sugar, and it will supply you with the sugar. So glycogen is a storage form of polysaccharide in animals. So it does has branches. It consists of a chain of alpha 1 to 4 linkages with alpha 1 to 6 linkages at the branch points. So this shows you the structure of amylopectin, and this is glycogen. So amylopectin is also quite branched, and similarly glycogen is also very highly branched. The next polysaccharide is chitin. Now, all the polysaccharides we have discussed till now, they're all called homopolysaccharides. So, why they called homopolysaccharides? They are the long chain of the monosaccharides, and they're made of the same monosaccharide. So, all same monosaccharide, like in starch, there is glucose, in glycogen also glucose. Even there is another polysaccharide called cellulose. Cellulose is also a homopolysaccharide because it's also made of it's also made by linking multiple glucose molecules, but the form of glucose is different. In cellulose, the form of glucose is beta form. So the next polysaccharide is chitin, and this chitin is basically also a homopolysaccharide because it's made of the same monosaccharide. It's a long chain made of the same monosaccharide. This is actually present in the exoskeleton of insects, and it is also present in the cell walls of some fungus. Now, cellulose, as I said, is made of beta form of glucose, and chitin's monomer is different. Its monomer is N-acetyl beta D glucosamine. So this is different from glucose because it has nitrogen in it. Okay, so this is N-acetyl glucosamine. This is a monomer of chitin. Okay, so multiple these kind of monomers linked together, they form chitin. Next, we are going to talk about heteropolysaccharides. Now, what are heteropolysaccharides? Heteropolysaccharides is a long chain of these monosaccharides linked together, but they are not made of one single monosaccharide like starch or cellulose or chitin, which we discussed. So these heteropolysaccharides, they are made of like different, they involve two or three monosaccharides. Okay, they're not made of one monosaccharide, they're made of more than one monosaccharides. Here is example we will be talking about. We are going to talk about the cell wall of bacteria. So the cell wall of bacteria is made of peptidoglycan. Now, what is peptidoglycan? So peptidoglycan has proteins in it as well as it has sugars in it. The sugar which is present in the peptidoglycan, which is component of the cell wall of the bacteria, has heteropolysaccharides in it. So the cell wall of bacteria is made of heteropolysaccharide and proteins, and the heteropolysaccharide is made of two different kinds of monosaccharides. One is N-acetyl muramic acid, another is N-acetyl glucosamine. So you can see here, like this represents the peptide. So this is this is N-acetyl glucosamine, and this light green is N-acetyl muramic acid. You can see there is N-acetyl glucosamine, then there is N-acetyl muramic acid, N-acetyl glucosamine, N-acetyl muramic acid. So the monosaccharides are two here, and they are crossed with some peptides. So this is peptidoglycan. So peptidoglycan forms the cell wall of bacteria, and peptidoglycan is made of heteropolysaccharides in the proteins. This is just a summary of polysaccharides. And the last thing we need to know about is what are glycoproteins. So glycoproteins are those proteins which has some sugar chains attached to them. The immunoglobulins, they are glycoproteins. So immunoglobulins are the ones which are part of our immune system. They are the ones which is the defense mechanism. Immunoglobulins are also called antibodies, and they are the ones which defend us against the infection. They are glycoproteins. Okay, that's a very, very typical example of glycoproteins. Immunoglobulins or antibodies, they are glycoproteins. And then these glycoproteins, they're also part of the cell membranes. We have talked about what are glycoproteins in when we talked about a cell membrane. And proteoglycans, they are basically present in the extracellular matrix of the animal cells. And proteoglycans are basically glycoproteins with high carbohydrate content in them. And that's all basically. And glycoproteins, as I said, they play a role in the immune response. And glycoproteins, basically, when they're present in the plasma membrane of the cells, they act as markers for the different cells. And these markers, which are basically made of glycoproteins, they are used in cell to cell interaction and also in the cell recognition. So that's all for this chapter. If you have any question, please let me know.